And now good transition to Gregory Fahy, who will speak about the reversal of epigenetic aging and immunosenescence trends in humans. Please.
All right, thank you. Let's get this going. Okay, so thank you very much. It's a pleasure to be here among so many talented investigators. And as you just heard, I'll be talking about the study that we published a couple of years ago, a year and a half ago. But I thought I would jump the gun a little bit and sort of preemptively move into the theme of this meeting, which we'll get into in more detail later. Does it really work? What reasons might we have to think that the results that we had really do work and are genuine?
Just a moment, Greg, do you want to share your screen? Do you have a presentation?
Oh, it's not working? Oh, okay. That's surprising. I already hit the share screen button. Are you seeing this now?
Starting, yes. Yes.
Okay. All right, let's go back to the beginning here and do it right. Okay, hold on. Okay, so I'm just turning off my cell phone here. Okay, so many of you are familiar with the study that we did. I just feel obligated to give a little bit of background about it. So our general idea was to do something about immune system aging as mediated by the thymus, and particularly by the atrophy of the thymus over time. So as you all know, the thymus is where we make our T cells, and they are major effectors of the adaptive immune system. And they are what is primarily affected by aging. So you see more changes in T cells than in B cells, for example. And the loss of competent T cells is related to thymic function, because since the thymus makes the T cells and that function peters out over time, you do expect to see some consequences of that. The consequences are delayed until after the age of 60, but then they become very significant. So the process of thymic involution is sort of diagrammed in the cartoon, showing that as we go from being a newborn to being 70, our thymus is replaced more and more progressively with fat, and both the cortex and the medullary volumes are gradually replaced by non-functional mass. And this results in major increases in age-related morbidity and mortality, and that's the reason that we chose to look at reversing thymic involution. And the other reason we wanted to look at this is that it can be done, it's achievable. There are many different approaches to thymic involution that have been investigated in the past, and I've listed most of these on the left. The one that appeals to me the most at the moment is growth hormone, or somatotropin, for several reasons. And one of those reasons has to do with the fact that it has a track record of being able to regrow the thymus in many different species, from rats to dogs to cats to humans, to HIV patients. In the cats, they did it with IGF-1, but of course, IGF-1 levels increase with growth hormone administration, so it's the equivalent experiment. And in these studies, by and large, not only does the morphology of the thymus come back with growth hormone administration, but also thymic function comes back in the form of the production of new cells, and even in some studies, improved function of the cells that are present. So this seemed awfully important for us to investigate. And people have been investigating thymic involution for many years, but everyone talks about it. No one actually does it for the most part. So we decided to actually do it. Now, I said I had several reasons to choose growth hormone among the many options available for study. One of the primary reasons is that, sort of like what Nir was talking about with metformin, growth hormone is not a mysterious drug. It has been approved for human adults since 1996 by the FDA. It has a good safety record. It does have some side effects, but they tend to be mild and reversible. And it has a number of additional benefits besides potentially regenerating the thymus, such as accelerating wound healing, improving cognition, regrowing cartilage, which is a big problem in and of itself. But the one that I'm showcasing in this slide is that aging is a catabolic process. And as we get older, the mass of many organs in the body declines, except for one organ, which is fat. So as we get older, the only thing that actually increases is fat. But Rudman showed many years ago in a 12-month trial that giving growth hormone actually regenerates not just thymuses. He actually did not look at thymuses, but other internal organs that are quite important for our health and well-being, such as muscles, such as livers, such as kidneys. I didn't show kidneys because it's not statistically significant, but we'll talk about kidneys in a while, and I think that what he observed is probably valid. So this is a profound anti-aging effect, and for that reason, that's one more reason that growth hormone was of interest. Now, growth hormone has been doubted by many people, and we just saw from Nir's presentation that there are correlations with higher rates of aging and higher IGF-1 levels. We'll talk about that a little bit more later, but I want to raise a point which nobody ever really talks about, and that is that growth hormone administration leads to an increase in insulin. And most people do not correct that. But since insulin is a highly pro-aging factor, what we decided to do is to negate that. And one of the ways that we did that is by using DHEA. And what you see on the left is a series of studies that I actually did on myself, in which I gave myself growth hormone for a week, saw an increase in insulin, and then and then at the same dose of growth hormone, added DHEA to the regimen, and it brought the insulin right back down to baseline. We verified that in TRIM, although we didn't really have time to go into that in the paper. We're also verifying that pretty profoundly in the TRIM-X trial, which is currently underway. So that's one reason that I wanted to try growth hormone, is that it does have side effects, but we can mitigate those side effects. And if there's not sufficient mitigation available from DHEA, then we're able to use metformin. And you've just heard the great properties of metformin that Nir just reviewed for you, many of which are quite relevant to what we want to do, including minimizing any cancer risk, which is not listed here, but Nir is right about that. I just want to point out in passing here that in the process of mitigating the side effect of growth hormone that I'm most concerned about, this hyperinsulinemic effect, we're using agents that in and of themselves have anti-aging effects. DHEA has been associated with lower all-cause mortality. In other words, low natural levels of DHEA or DHES are associated with higher all-cause mortality, frailty, cardiovascular disease, mortality, stroke severity, the risk of going into a nursing home or a hospital, increased arterial stiffness and inflammation. And you've just heard of many benefits of metformin. So by using these agents to counteract the side effect of growth hormone, we may unmask the benefits of growth hormone, as well as gaining additional benefits from these other two agents, which is an important factor for later on in the talk. So based on all of this, we did the TRIM trial. It was ended up being a 2-year trial. Each person in the group was treated for 1 year, but we couldn't sign up everybody in the group simultaneously, so we had 2 cohorts. You see the first cohort of 6 in the middle, and then on the right you see the last cohort of 3 guys, all successfully with their graduation Thymonaut t-shirts on, or jerseys, I would say. And so the aim of the trial was to regrow the human thymus using a combination of growth hormone, DHEA, and metformin. We had 9 healthy men aged 51 to 65. We treated them 4 times a week for 1 year. We adjusted the doses for each person in the trial based on their own individual response to try to optimize their treatment. This is based at Stanford. We were able to use the Stanford Human Immune Monitoring Center to do a lot of our immune system analysis. The paper was published in September of 2019. It's an open access paper, anyone can read it. So just to quickly highlight the study results, the main thrust of the study, of course, was to look at immune system aging. We did see thymus regeneration. We documented that pictorially and also statistically. The p-value was 10 to the minus 18th, so we're pretty confident we did regenerate the thymus. Interestingly, in looking at the thymus, we also looked at the sternum, which in images that we collected is right above the thymus. And we also showed that the adiposity of the sternal bone marrow was also decreased, and that may have other implications. Getting to the immune system results, we did see an increase in naive T-cell populations and also in recent thymic immigrants. And we also saw, interestingly, since we were very concerned about the possible risk of cancer, we saw what looked to us like indications of a reduced cancer risk. PD-1 signaling on exhausted CD8 cells went down. Indices of prostate cancer risk went down. And something called the lymphocyte to monocyte ratio, which has been correlated with less severe effects of at least 19 different kinds of cancer, went in the right direction. It went up. At the same time, inflammation, age-related inflammation, presumably went down. And with respect to one of our goals of controlling insulin, insulin was at least partly controlled. As you can see at the red arrow, we actually did reduce insulin at one point, even though we were increasing the growth hormone doses at that time. We did have one guy who had a poor insulin response at the end, so we didn't end up at the end as well as we should have without him, but we certainly controlled insulin a lot better than we would have without trying to mitigate that side effect. Now, in the course of doing the study, we also saw some things that we were not really expecting. One of the most dramatic was a restoration of hair color in some of the volunteers. This is one example, the only one that we could really document properly, although I did find some more photographs recently, so I will see if we can assemble another example for you of that effect. We also saw increase in renal function, at least estimated GFR. And in this diagram, the treatment ended at 365 days, but we were able to do follow-ups on 6 of the thyminots 6 months later, and it seemed like the trend might still be continuing, although we didn't have enough people to prove statistical significance at that point. The other thing that we saw that I think may be significant is that we saw a decline in both total monocytes and CD38 positive monocytes. And that may be really significant because monocytes tend to drive down tissue NAD levels with age. That seems to be the main mechanism by which we lose NAD with age. It's due to dissolution of NAD through this ectoenzyme CD38, strange as that may seem. So that may relate to the last result we observed, which is the most surprising of all, which is across the board with 4 different epigenetic aging clocks. We saw a highly significant in each case, reduction in epigenetic age relative to where you started from. So we got rid of a lot of noise in the background statistics by subtracting out the offset in epigenetic age before treatment. So the offset is the difference between the epigenetic age of the person and their biological age at baseline, which is time 0 before we started treatment. And then we just subtracted that standard difference, that starting difference, from that difference as it evolved over time, so that we could compare everybody on the same basis. So everybody shows a 0 change in epigenetic aging at time 0, and then everything else is where they change relative to themselves. So each person served as his own control in this study. And you can see that even though the clocks are composed differently and measure things differently, they are unanimous in showing the same overall trends. And if you average them, you see the statistical significance is there for all time points in the trial. And also you can see in all of these cases, but certainly in the average, that the rate of change of epigenetic age seemed to accelerate after 9 months. So from 0 to 9 months, the rate of epigenetic aging reversal was about 1.6 years per year, or 1.6 days per day. And in the last 3 months, the aging reversal, as judged by the clocks at least, was about 6.5 days per day of treatment. So now we get down to the kind of theme of this meeting, which is, can we believe any of this? You know, it looks like it worked based on what we saw, but could it just be a fluke or some flaw in the way we analyze lack of a control group? The only way we controlled the study was by using each individual as his own control. Maybe other people not treated would show the same changes in theory. So are there any other reasons that would allow us to decide whether it's reasonable that this worked, that it should work, that we should have some confidence that this may really be a real effect? And one of the bits of evidence along these lines is the fact that there has been previously demonstrated in a series of obscure papers a link between the thymus and aging on a generalized basis. So if you give thymus transplants to old mice, you see that relative to the youthful value, so in the figure, that's 100% of young control value, you see a restoration of beta receptor signaling in the submandibular gland. You see reversal of hypothyroidism. You see an improved population of brain beta receptors. You see restored insulin sensitivity as indicated indirectly by lowered plasma insulin levels, and you see reduced liver polyploidy. Now, I can't explain to you why you should see any of those things based on correcting an immune system decline, but as Josh was saying, aging is a network. Everything talks to everything else. Everything in biology happens for a reason, in my opinion. And so the immune system may have a lot to do with being one of the pacemakers of aging in general. Now, one of the things that I need to point out is that we did try to correlate the epigenetic aging reversal that we saw with changes in fat content of the thymus. And we did not find a correlation. So we think that the epigenetic aging reversal may be independent of what happens to the immune system, at least on some level. Probably they both contribute to the aging reversal that we saw, but there may be other effects that are non-immunological. And one of those immunological effects may be related to NAD. So as I pointed out before, monocytes in general and CD38 monocytes in particular decline. And you can see we achieved high statistical significance at 9, 12, and 18 months. So even after the end of the trial, 6 months later, the CD38 monocyte population remains, significantly depressed. And since these cells may drive the decline in NAD, and since reversal of NAD decline has been linked to many signs of aging reversal, at least in mice, and those are some examples from 2 of the early papers from Gomez, so that's the Sinclair lab, and then later some Zhang's lab and Science. That provides a rationale for why our treatment, one of the reasons why our treatment may have had an effect on epigenetic aging and other signs of aging as well. That if we restore mitochondrial function, reduce inflammation, etc., we may expect to see this show up in the epigenetic aging clock results as well. The other consideration is one of the ones I brought up before, is that each one of these agents in our cocktail is independently shown to oppose aging in some way. So aging is catabolic, growth hormone is anabolic, but I've already run through the anti-aging correlations for metformin and DHEA as well. So as Josh was saying, and I agree with him, aging is a multifaceted process. You're not going to affect it as profoundly as you might by just using one intervention. These three interventions definitely impact complementary and major, massively important pathways that relate to aging. And so it makes, it's plausible to me at least that they should be expected to have the kinds of results that we saw, or at least it's plausible that they would. So one last consideration, this goes to what we heard from Irina and from Harold, is just to point out that DHEA and growth hormone are both present in young animals. And we've heard about, Harold's observations about young plasma factors. And I would also make just an observation about Irina's comments that although in her studies it was sufficient to reduce old age factors, the pathway through which that was beneficial was by rebooting the youth factors, and it's therefore possible that giving youth factors would also be beneficial. In any case, I can't say that DHEA and growth hormone would be able to match anything like what Harold's cocktail has, but they are present in youth, and so it's worth a thought that this kind of, what we observed is kind of consistent with the parabiosis study, what Irina and Harold have observed in the past. I just have a couple of other things. This is the last real slide in the show. We found high statistical significance in many of the parameters in our study. They were all self-consistent. So we saw improvements not in just naive CD8 cells, but also naive CD4 cells, et cetera, et cetera. And it's hard to have bogus results achieve high statistical significance with just a handful of people. It's very, very improbable. So it makes me think that we'll be able to replicate these studies as we go forward. We also had a different way of estimating biological age that came up just in the last few months, which used a plasma factor way of estimating biological age, essentially. Things like insulin, other things that you can readily measure in plasma to estimate biological age, and we got the same result using that we did with the Horvath clocks. Regression of 2.5 years in the TRIM study, retrospectively observed. That strengthens the evidence that actually this treatment does work. And there may be other evidence. I started a pilot version of this myself early last year, and noticed that my intraocular pressure went down. It's kind of weird, but there may be other evidence that we can deduce to show that this actually may be a real effect. And I just will make one last comment about the IGF-1 story. So every good gerontologist believes that I'm nuts because IGF-1 is terrible and growth hormone is horrible and everybody dies much more rapidly if you have more of these things, but actually it's not really true. So in the centenarian studies, you have to understand the effects are small, and in the centenarian studies, as well as in most mouse models that have promulgated this idea, growth hormone and IGF-1 signaling have been altered during gestation and in the prepubescent state. And for example, in the Ames mouse model, as you can see at the bottom, at 18 months in controls, you have an inflamed hypothalamus and the Ames mutation, which knocks out growth hormone IGF-1 signaling. You don't. But in the Ames model, if you give growth hormone before puberty, you abrogate the mutation effect and you get age-related brain inflammation later in life. But if you wait until after puberty and give growth hormone, there's no detrimental effect. So the effects of growth hormone IGF-1 signaling, I think, in adults are quite different than they are in young animals. And therefore, one reason to sort of disbelieve that our treatment could actually have a benefit. May not be as strong as people tend to think. I would just put it that way. So I think the treatment probably works, but we are doing an extension trial, the TRIM-X trial, to see if we can replicate the study results. We will include women, we'll include people of greater ages and also people of younger ages. And many people have come to us wanting to help us understand our results by running assays for us on our blood samples that we are not able to run. We're certainly quite happy to collaborate with anyone who would like to do that. And that's how to find me. I hope that perhaps we can definitely show in the future that this treatment does work.
Thank you very much, Greg. We went a little over time.